There are some in science who are mistrustful of imaging techniques, and I think any imager would be at the top of that list. Imaging people know full well how much these techniques depend on statistical analysis and how easy it is to make a mistake in your assigning of groups or your statistical methods. And so I would first want to reassure anyone interested in the technique that it is robust to statistical multiple comparisons testing. It's robust to every type of control study we've thrown at it. Is it a perfect technique? Absolutely not. One of the major limitations is that the size of voxels in MRI is larger than the size of striosomes, and therefore we're able to say a given voxel behaves purely like a matrix or a combination of striosome and matrix. We're not able to image at a fine-grained enough nature to say it's purely striosomes. But with a higher field, perhaps at 7 Tesla diffusion imaging, we could get to that. This is purely a starting point. The technique will get better over time. I'm confident that with higher level MRIs, with better statistical approaches, we could make the technique even better. So at this point, this is purely a proof of concept presentation. It's all well and good for, I have 3 independent imaging cohorts. I have 123 subjects. It's a very large sample size, but it's one study so far. This hasn't been adopted in any trials so far, but we're looking for collaborators who'd be interested in using the technique in their own study populations. One of the potential applications would be in individuals who carry the Huntington gene and who we can predict when they will get Huntington's disease. This technique might be useful to be able to track longitudinally when you start to see cellular pathology and find out whether that's something that correlates with the clinical changes of Huntington. I think the most important thing that this technique would allow people to do is simply to think in a much more fine-grained manner about striatal injury. When we look at an MRI and see that someone has had a stroke or someone has gliosis affecting their striatum, we think about this as a sort of a unipolar sort of injury. It's just hit the striatum. This technique will allow us to actually think much more granularly to say this is a more matrix-specific injury or striosome-specific injury, and therefore we would expect to see different phenotypes arise from those. These are very distinct types of neurons. It's just that so far we haven't been able to tell them apart until the person had died and we use histology. So this technique allows us to take one step farther back in the mechanistic cascade. One of the major limitations to movement disorders therapy now is that our biggest gun, deep brain stimulation, really has only one target for deep brain stimulation. That's the globus pallidus interna. We know that the GPI has subfields within it, and it may be the case that different dystonia populations respond better to stimulation of different parts of the GPI. It may also be possible to say a given dystonia population will respond better to GPI plus another target, or a different target entirely than the traditional GPI target. As the best we can do at characterizing the structural injury leading to dystonia, the better we'll be able to stratify dystonia populations and say who should get the traditional DBS versus who should get maybe a different approach and what that different approach might be. Those are purely theoretical at this point, but it's easy to imagine that a better anatomical knowledge would allow us to do better stimulation and better outcomes.